Okay, so welcome to this session. In this session, we are going to discuss, and through discussion, we are going to learn cell as the fundamental unit of life. So this record is created with an intention that whenever you get a chance and opportunity, you would be able to revise and learn through that. Okay, so spending some time to talk about few fundamental concepts that anybody requires. First, let us understand what we have learned before. Previously, we have learned what is cell, right? So what is a cell?
A cell is a basic, fundamental, and structural unit.
We say that cell is a basic structural and functional unit of life. What do you mean by structural unit?
Which structures anything, any object or
Anything, yeah, anything that is structured with the help of the cell. When we say anything, we mean any living organism. So you know that there are 13 properties of life. So if there are 13 characteristics generally which are shown by all the living organisms, and out of those 13 one point is cellular structure. Every living being shows a cellular structure, whether it could be unicellular or whether it could be multicellular, right? So that means any organism that we have around us will be created out of cells. If could be one or it could be many. So that is why cell is the structural unit of life. It is also called as the functional unit of life. Why?
because it in at minimal level it carries out many functions.
At a minimal level, it carries out all the functions, most of the functions that is carried by a complete living organism. So that is why it is the functional unit of life. Okay. Have you been introduced about cell organelles? Do you know? Okay. Let us learn what are we going to learn this. We are going to understand what is a cell, the difference between plant cell and animal cell. Then we are going to understand the structure of different cell organelles and the function. Here is a graphical drawing of a cell. You see that it looks like a transparent ball in which there is something in the middle. This middle part is always known as nucleus. And the next part, which looks like clear water, like a gel, we will call it to be cytoplasm. And then we have a membrane around it, that is the limit, that is the border. We call it as cell membrane. So we will focus right now on cell, but before that we shall go into some basics which we should learn and should have learned before. First, living beings could be living in any environment, and different organs in a living organism performs a different function. In the same manner, cell has different organelles. Clear? What is organelle? Organelle is a part of the cell which performs one specific function. Performs one or a few specific function. Already we have understood that is why cell is the functional unit of life. Means if an adult body does ingestion, cell also does ingestion. If an adult body does digestion, cell also does digestion. If an adult body, full body, multicellular body does respiration, cell also performs respiration. The study of cell is called as
Cellular biology.
cell biology, not cellular. Cell biology, otherwise known as cytology.
Cytology.
Actually, these two are terms which are created one after another. Initially, it was called as cytology, but which studied the cell. But then we could know that cells also have organelles who function. So if you ask, cell biology is a bigger subject than cytology. Cytology tries to study only cell. When it originated, it tried to study the cell structure only. But cell biology is a bigger subject. How do you study cell? It would not have been possible if we do not have discovered something called as a microscope. because human eye has a limitation of vision. So invention of microscope was a milestone. It was a milestone because it helped people to see. Okay, go and open that. So you know very well that microscopes for the first time were designed by Antony von Leeuwenhoek, A.V. Leeuwenhoek. Magnifying glasses had been used since long. The tailors were using it, the watchmakers were using it, anyone, the jewelers were making it. But Antony von Leeuwenhoek, you know, was a tailor, was a draper. So as a piece of work, the drapers did something what is called as mending. So where they join the threads. So for that you need lenses. So lenses. So it was an accidental discovery that he could know that the combination of lenses can help somebody to see something closely. And Antony von Leeuwenhoek lived from 1632 to 1723. He was a draper, and since he started to discover things which were not seen before, right, with the help of microscope, he is also became a founder of microscopy. So he was a scientist. So what he did actually, if you see, yeah, this is the work of Antony von Leeuwenhoek. It is a simple microscope. What do you mean by a simple microscope? A simple microscope is a microscope which uses only one glass to magnify. Clear? So this, with the help of this simple microscope, he started to examine various things. It was a single biconvex lens. Biconvex means convex from both the sides. And it had a considerable magnifying ability. And with that, he could see that in a drop of water, there were some moving creatures. Since at that point of time, movement was called animus. Animus means movement. So he named them animalcules. He did not know what is that. Since they were small and they could move, he called them to be animalcules. But for the first time, he was observing microorganisms. Now we had some organisms. Now the world came to know that there is an organism which is so small that it cannot be seen through naked eyes. Because previously, people used to think that diseases are caused by foul air because they didn't understand there is a bacteria, there is a virus. They never understood that. However, that is why Antony van Leeuwenhoek is also called as the father of microbiology. Microbiology. So the branch of biology which studies
Microorganisms.
right. So now here is a timeline of inventions. There is a famous company called Janssen. This is named after Zacharias Janssen and his father Hans. So Janssen and Hans today also is a very famous lens making company. You go to any optical shop where they make lenses, they may ask you that do you want to use Hansen Janssen lenses? It was a father and son company which during was established in 1590s. In 1590s, they mounted two lenses in a tube, the first compound microscope. Clear? In 1609, it's only 19 years after, Galileo Galilei constructed a compound microscope with a convex and a concave lens together. Convex and concave lens together. How these principles work, you will study in physics when you study optics. Now, in 1674, that means approximately how many years? 65 years after, A.V. Leeuwenhoek constructed an extremely simple instrument. It only had a single lens mounted on a tiny hole in a brass plate, and through this simple microscope he was able to observe microorganisms. Okay, so who else were trying? Just 10 years before, like it is 74, so 9 years before, there was another person. He was an Englishman. His name was Robert Hooke. This English scientist in 1665, he started taking the slices of cork. What is a cork? Cork is a part of a plant. It is the part of a bark of a plant. It is in the inner side of the plant. This is spongy. Nowadays we are using plastic caps, but then the corks were used to cap any bottle. Homeopathic, champagne, wine, they came with cork. So there used to be also an instrument called as corkscrew. So you turn that instrument like a screw and pull it, the cork opens. So that is a cork. Corks were also used when I was a child, they were used in making boats. However, they are not found much today. So he took cork and took a very thin slice, and he observed it under his designed microscope, which you can see seems like a compound microscope. Yes, it was a compound microscope. When he saw that compound microscope, he saw pictures like this. It looked like some chambers, okay, hollow chambers. So he called them, these box-like compartments, box-like chambers, as cell, because cell is the Greek word for chamber. But he said cell is the basic unit of life, because he believed that anything, mostly plants, because he had only experimented with plant, so he believed that these cells constitute the plant body. But if it comes to moving things, the discoverer was A.V. Anthony von Leeuwenhoek. Up to now you are clear? Now let's coming to what is an ordinary compound microscope. Well, a compound microscope is an optical instrument which consists of two convex lenses of very short focal length. Short focal length means the focus should be nearer.
Much nearer to the sun.
So what type of convex lenses have a focal length short? Is it more flat or more spherical?
More flat.
No. Remember, if the refraction is more, turning of the ray, light ray is more, focal point is closer. Clear? So if a lens is like this, another lens is like this one will have a shorter focal length. So that means these lenses are more spherical. I hope you understand. So he used two convex lenses of short focal length, and the compound microscope could magnify anything up to magnify the object up to 1000 times. 10 to the power 3 times. So that means you can see anything which is 10 to the power minus 3. I hope you understand. Well, what is the principle? The compound microscope works on the principle that when a tiny object is placed just beyond the focus of the objective lens, acha, what do you mean by objective lens? In a microscope, there are two major lenses. One is an eyepiece in which you keep your eye. Another one faces the object, that is called objective lens. And the idea is that if you keep the object just beyond the focus of the objective lens, whatever is the focal length, उससे थोड़ा आगे रखोगे, a virtual, inverted and highly magnified image is formed. How it is formed, you will study in physics actually.
If it is
Get just after focus, a virtual image. Virtual means it cannot be projected, it can only be seen. Clear? Inverted and highly magnified image will appear. Understand?
okay. But if you make it to the directly to the focus, it will become
Blurry. We'll talk about that. This is the practical part, how it is adjusted. Okay.
Okay.
So, and at least the distance of the distinct vision from the eye held close to the eyepiece. You have to keep your eye close to the eyepiece in order to be able to see that. So the length of the tube, how far will be the object, all these are planned, and accordingly this compound microscope is created. But compound microscopes also have limitations. They can magnify things up to some thousand numbers of times depending upon the quality of the objective lens. Like some objective lens come up to the magnification of 10,000, 2,000, 4,000. So more expensive the microscope, better would be the visibility because the objective lens would be different. However, you cannot go beyond 10,000 or 20,000 times. But when you are trying to study things even smaller than the cell size, cell is small and the organelles are even smaller and the atoms and molecules are even smaller. So this technique didn't work to go for a high magnification. So at a result, there were microscopes called as electron microscopes which are designed. An electron microscope can magnify something 2 lakh times. 2 lakh times. So with which you can almost see any atom, a crystal. Are you getting my point? Also structures in the cell. So today electron microscopy is used. How does it work? It works by throwing an electron beam. If you have ever seen any X-ray machine, an X-ray machine throws rays, yeah, and the rays pass through your body, and the rays are captured by another photographic film, and there you can see the image. Similar technique is a ray of electrons is put through a lens system, okay, through a lens system. Since it is more precise, it can bring magnification and whatever image is created, that is being presented on a computer. Understood? Now, so you see that if we are able to study cells today, we are able to do that because there have been many people who have contributed. At least some names you should know for Olympiad's purpose and for the purposes of your understanding, knowing. Zacharias Janssen, I told you, with his father Hans, invented first compound microscope. He was an optician, lens maker. Then Robert Hooke, 1665, discovered cell from cork. A.V. Leeuwenhoek, 1674, first observed living cells of bacteria. Sperm is the male gamete present in human ejaculated liquid. Protozoa is one cell animal that is free living. Okay, It does all the functions, but it is free living in water. Also, any watery environment. Then you had Robert Brown. Robert Brown could for the first time tell that there is a presence of nucleus inside the cell. Previously, everybody used to think that how does the cell work? Now we could know that there is something in the middle which is like the brain of the cell. So the credit goes to Robert Brown. But it happened in 1831. So approximately how many years? Approximately 130 years, for 50 years. 150 years after, you get to see that there is a nucleus. Another name to be remembered is Johannes Purkinje. Purkinje is remembered for two things. He demonstrated that there is protoplasm. So what is cytoplasm and what is protoplasm? The clear liquid around the nucleus is called cytoplasm. Cytoplasm plus organelle is called as protoplasm. Bujhiyala? So protoplasm gave us understanding that we have many organelles which are even smaller than the cell working inside the cell. Clear? And he is also credited for discovering cell division. Means by this point of time they could know that living cells can divide. But how do they divide? How that protoplasm gets shared? How does that nucleus get shared? He could discover it because there are very thin fibers which start from centrioles and connect to the chromosome and pull them. That's why these are called as Purkinje's fibers, named after him. Okay, you will study that when you study cell division. You should remember Camillo Golgi, who for the first time discovered something called as Golgi complex. This Golgi complex is responsible for restructuring the protein. Reticulum synthesizes protein. Golgi redesigns the protein. We will study about that later. We must remember Palade, George Emil Palade. He talked about ribosome, which is the tiniest organelle inside the cell. Even there are but they are not full organelle. Ribosome is the tiniest. Then you have
Ribosome?
huh,
Oh, it's not the same as rhizobium.
no, rhizobium is the name of a bacteria. Ribosome is organelle. Then comes Benda, Carl Benda. She coined the term mitochondria. Mitochondria is otherwise known as the powerhouse of the cell, the energy producing part, organelle. So, like this, many people kept on giving understanding, better and better understanding regarding cell. Yes, what is the doubt?
Cell organelles are the parts of the cell which are responsible for functioning the cell.
For carrying out the different living functions, life functions of the cell, inside the cell. So, after all this understanding, a theory came out. Means it's not like that if one happened next, it was happening simultaneously. People, scientists were able to understand in a better manner. So we had something called as cell theory. This cell theory is proposed primarily by two scientists initially. In 1838, there was Sleeden. Sleeden was German and he was a botanist. Okay, botanist, study plants. So full name is Matthias Jakob Sleeden, M.J. Sleeden. He told that cell is the building block of all plants. उन्होंने कहा हर कोई प्लांट जो भी आप देखो वो सेल से बनता है. There was another German physiologist. Who's a physiologist? Physiologist is a person who studies the functioning of the human body. How does human body function? So there was a physiologist German. His name was Theodor Schwann, T. Schwann. तो पहले थे M.J. Slidin 1838, 1839 T. Schwann. He told that cells are the fundamental units of all animals. So one person said, confirmly, prove it. Theory, you understand? Theory is something that cannot be rejected. Okay, so he proved it ultimately that every plant is made of cell. Schwann proved that every animal is made of cell. So from this we got two fundamental propositions. What is that? All living things are made of cells. That is the proposition number one, statement number one. After few years, means another 16 years, there was another scientist. His name was Rodolph Virchow. He was also German, but he was a physician, he was a doctor, and he was an anthropologist who studies human evolution. So he came, the third statement, which was in Latin. Latin, you know, the language of Italy, Rome. Omnis cellula est cellula. That means every cell comes from pre-existing cell. हर सेल एक पुराने सेल से बनता है, बुझेगा? By division of the cells. Omnis cellula est cellula. So in this way, we had basic three axioms, three statements of cell theory. वो क्या है postulates? Postulate मतलब statements. One, all living organisms are made of cell. So cell is the structural unit. Number two, cell is the fundamental or basic unit of living organism. Right? Third, all cells arise from pre-existing cells. Understood? Well, let us take the example, explanation with a rose plant to understand. Take any part of the rose plant, see it under microscope, and you will always find that there are cells. So structurally, it is made of cells. Cell is also functional unit of life because in rose plant, food is prepared through the process of photosynthesis done by cell. तमे देखो जो सेल को मैग्निफाई करके एठी देखा होची, here you see some green color organelles गारा गारा हेकी पड़ची. These are called chloroplast. They contain chlorophyll. So if the plant is making food, it is inside the cell. Similarly, absorption of water is done by the root. किंतु ultimately सेटा कोठी की जाउची. ये जो water mineral असला से जाके कोठी पहुंचुची. Cell. So cell is also observing. Whatever the waste material is generated that comes out of the cell. So any activity which is done by a living organism is also done by the cell. That is why it is the functional unit. And omnis cellula is cellular. Jetebede ame observe kolu cell gudaku continuously that they keep on dividing to make new cells, and the new cells grow and then they die. Okay, after working. So like this, cell theory postulates were proven to be correct. Understood? Now, what's the doubt?
Functional unit ଆମେ ଜାଣିପାରୁଛୁ cell
हाँ, सेल ऑलमोस्ट सबू करती. तुम कहि परबनी एग्जैक्टली सबू करती कारण फंक्शन गुडाक स्पेशलाइज हुए. अब गोटे मल्टीसेलुलर ऑर्गेनिज्म रे गोड़ लोकोमोशन करूची, किंतु तमर कोन हार्ट रो सेल गुडाक लोकोमोशन करछन्ती तो करिवे तो हार्ट को छाडी पड़ेबे, खन 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 पड़ेबे. बुझि परछो तो? सो स्ट्रक्चर्स सम फंक्शंस, नॉट ऑल फंक्शंस. बुझिला? एबे देखिबा. So Bartaman ame take away kon naba from cell theory that all organisms are composed of cells. Number two, cell is the structural and functional unit of life. Right? Third number, every organism starts its life from a single cell. गोटिए cell रू सबु organism आरंभ हुवंती, unicellular हो कि multicellular. No cell arises spontaneously. माने बिना किछि रे गोटे माने किछटा प्रोटीन किछटा फैट एमती मिसी मासी के सेल तयारी हुएनी. बुझिपारछ तो हाँ it should be created before and develops into organisms. All cells arise from pre-existing cell. Okay, next, all cells are basically alike in their chemical composition and metabolic activity. So एतिके समान कि सेल विभिन्न काम करथै पारे किंतु ता भितरे जो मेटाबॉलिक प्रोसेसेस हुए. व्हाट डू यू मीन बाय मेटाबॉलिज्म? The basic process of anabolism and catabolism together. Anabolism माने synthetic processes, catabolism मतलब breakdown processes. बुझेला? बॉडी रे सब बेले सिंथेसिस चलता है, जिनसे तैयारी होता है और जिनसे भांगुता है. टुगेदर इट इज कॉल्ड मेटाबॉलिज्म. और मेटाबॉलिज्म जोगु लाइफ चले. बुझेला? फूड स्टोर हबो, सेलटा बढ़िबो, सेल पुनी भांगिबो, सेल काम करिबो, डिस्ट्रॉयड हबो, तू बंची की रहबो, मु बंची की रहबे. सो दिस इज व्हाट इज मेटाबॉलिज्म. सेल रे सब सेल गुड़ाकर बेसिक मेटाबॉलिक एक्टिविटी समान. से माने किछटा रिसोर्स नियंती, किछटाकू रेस्पायर करंती, रेस्पिरेशन करंती, एक्सक्रीशन करंती, ए गुड़ाक समान. बुझिला? नेक्स्ट, the functions of an organism as a whole correspond to the functions carried out by itself. तू जहाँ भी करिबो तमे as a multicellular organism, एटा function of the cell. बुझिला? तमने if I am removing carbon dioxide by respiration, by breathing out, the carbon dioxide had originated from cell. They have been collected and then they have been thrown away. बुझिपारछो? If the body is producing urine, the urine has been collected. The urea has been collected from the cells. बुझिपारछो त म कोन कहूची? So ultimately the functions which you carry out, life functions, that is the output of the functions carried by cells. बुझिला? एटा पड़े परीक्षा रे, सेनु एटा को मने रखियो. ओके, मु नोट दे देबी वीडियो रिकॉर्ड, बट मने रखियो, रिवाइज करिवो, नोट करिवो. तापरे आमे आसीबा मॉर्फोलॉजी एंड इंटरनल स्ट्रक्चर ऑफ द सेल. एबे आमे बुझिबा कि व्हाट इज मॉर्फोलॉजी. मॉर्फोलॉजी इज द स्टडी फ्रॉम आउटसाइड. गोटे जिंस बाहरु कोन दिसे ताको आमे मॉर्फोलॉजी बोली कहू. बुझिपारछ तो? What it appears from outside. बुझिला? So and internal structure माने सेल भितरे कौन अच्छी. Anatomy एठी सेल लेवल रे हमें कहूना. Anatomy is a term which is kept for full multicellular organisms. तेणु हमें यूज करिबा internal structure. एबे हमें देखिबा कि the cells shape, size, structure they differ. काहे कि differ करे? It differs because of the complexity of the function and the type of organism. प्रत्येक organism रो आवश्यकता अलगा. अमीबा गोटे सेल भितरे सब काम करनोछ, किंतु तमरो विभिन्न प्रकार जिंस दरकार पड़छ. तमरो पार्टी दरकार पड़छ, ओके, नाक दरकार पड़छ. सो सेल गुडाक मॉडिफाई करनियंती निजको अलगा अलगा सिचुएशन रे, अलगा अलगा ऑर्गेनिज्म रे. बुझिपारछ त? So that they can make the basic structure of the organism and do the function. नाकरे भी सेल अच्छी, आखिरे भी सेल अच्छी, किंतु तमे कहि परिबनी आखिर सेल जहा नाकर सेल से. बेसिक फंक्शन समान, किंतु there is a structural differentiation. बुझेला? हेयर रे भी सेल अच्छी. बुझिपारछ? आउ नख रे भी सेल अच्छी. तामाने कोन हेयर इज इक्वल टू नख, स्किन इज इक्वल टू आईबॉल. बुझिपारछ? सो ए कॉन्सेप्ट को हमें बुझिबा. एटा को हमें कहू सेलुलर डिफरेंशिएशन. सेलुलर differentiation. What do you mean by cellular differentiation? A cell becoming different in a different environment. हमें कहलु ना every living being starts from a cell. गोटे सेल रो स्टार्ट हुए. ताल तो तमे सेही भलिया गोटे बड़ सेल टे की बंची थांत. तमर आखि नाक गोड़ पाद ए सब केमती हेला सेही सेलटा प्रथमे मेंचे डिवाइड कला डिवाइड करके से किसी समय पर पुणी बदली गला तार स्ट्रक्चर बदलला तार शेप बदलला ठीक तुम जहाटा पाइल सेटा होची गोटे डिफरेंट स्ट्रक्चर बुझी पाछ बुझी पाले पर्यंत केमती हुए परे पड़ो नॉट नाउ एबे हमें बुझिबा व्हाट डज इट मीन बाय यूनिसेलुलर एंड मल्टीसेलुलर ऑर्गेनिज्म नेम इंडिकेट्स if it is a unicellular organism it consists of one cell ta mane all the basic life processes reproduction okay nutrition how kon life processes guda ka basic digestion respiration control and coordination control coordination nathile cell will die cell jodi decide karibani ko jinsa ta mate nabar achi right if the cell does not decide a cell doesn't decide what thing needs to be taken the cell may take toxins बुझिला cell decide करिवो केते बेले को प्रोटीन टा तयारी करिवार अछि so control and coordination is also done by the cell internally so ए भली सब जाक जतिक कॉम्प्लेक्स फंक्शन जहा कि हमें सेति पई मु तुमको कहछि लाइफ फंक्शन टा को रिवाइज करू जो लाइफ प्रोसेसेस देछि ना प्रिंट आउट टे ताको रिवाइज करे पे सेति देछि काहे कि बिकॉज तुमको आउथरे रिविजिट करिया दरकार व्हाट आर द लाइफ प्रोसेसेस तो जो कॉम्प्लेक्स मल्टीसेलुलर फंक्शन गुडा को गोटे मल्टीसेलुलर ऑर्गेनिज्म करे गोटे सेल मध्य यूनिसेलुलर सेल मध्य सेतिकी करे हैला सो से यूनिसेलुलर कोटा यूनिसेलुलर ऑर्गेनिज्म मूवमेंट करे जदि यूनिसेलुलर थिबो कारण मूव न कले फूड कोड पाइबो लोकोमोशन भी करे कारण लोकोमोशन न कले खाद्य सर्च केमति करे let us see even a bacteria don't you think bacteria move then how does it grow बुझि पाछो तो the bacteria also moves with the help of cilia तार सेल रो सरफेस ऊपर हेयर लाइक छोट छोट स्ट्रक्चर थाए सेटरे से चले से सेल रो सेल वॉल रु तयारी थाए सेल मेम्ब्रेन भितरु सेटा बाहर थाए किंतु सेटा ताको चलीबारे हेल्प करे amoeba good example से लोकोमोशन शो करे तार सेल मेम्ब्रेन टा को ही से गोड़ा कर दे सेल मेम्ब्रेन टा को छुई दबो ता बॉडी टा को आगु टान नबो दिस इज हाउ इट मूव्स सो कहबार कथा होची कि यूनिसेलुलर ऑर्गेनिजम्स मानकर अनेक गुडिये जनरल फंक्शन गोटिए सेल रे हे जाय बट डिपेंडिंग जतबेले हम ताको माइक्रोस्कोप तळे देखु डिपेंडिंग अपॉन द न्यूक्लियस ताको हम दीटा कैटेगरी रे डिवाइड करू eukaryotic prokaryotic the word caryon karyon रो माने होची न्यूक्लियस सो prokaryotic मतलब primitive nucleus माने एठी न्यूक्लियस रे गोटे प्रॉपर शेप न थाए doesn't have a proper shape and when you talk about prokaryote the material inside the nucleus what is there inside the nucleus now we know we have chromosome dna rna all these things so this dna or chromosome is present like a jelly ball of noodles you know that area you can see but it doesn't have a ball a membrane a coating so we in case of prokaryotes call that as a nucleoid not nucleus the term is used for
nuclear means
which has a shape and also membrane that membrane is called membrane matlab coating covering called nuclear membrane which binds the dna chromosomes inside bujhila so what are the examples of prokaryotes these prokaryotes are primarily bacteria bujhila okay kintu eukaryote they have a full formed nucleus and if you see there are many differences तो हमें कौन करिबा टिके differences को analysis करिबा first let us analyze the difference between unicellular and multicellular organism. Unicellular has one cell, multicellular has many cells. Unicellular, all the functions are performed by one cell, but in case of a multicellular organism, different cells perform different functions, which I told you, it is a cellular differentiation. The cell गुड़ाक differ करके अलगा हेके सेमाने तैयारी करंती टिश्यू गुड़ाक मिसिकी करंती organ, and organ काम करिबा आरंभ करे, बुझियला. Third, in unicellular organism, division of labor is absent. अब से तो सब काम निजे ही करूचे. हाँ, ऑर्गेनेलिज थाई पारे, किंतु सेठी प्लाज्मा मेम्ब्रेन फूड को रिसीव करिवो, प्लाज्मा मेम्ब्रेन पानी को भी नबो, प्लाज्मा मेम्ब्रेन एक्सक्रीशन भी करिवो. The outer membrane of the cell, I told you. Cytoplasm is the liquid. Plasma membrane is the membrane around it. Cell membrane. So a cell membrane पानी को रिसीव करिवो, पानी को छाड़िवो, दरकार पड़ले चलिवो अमीबारे, पुणी केते बेले दरकार कले एक्सक्रीशन करिवो, वेस्ट को रिमूव करिवो. तो कोन तार स्पेशलाइजेशन अछि. So there is no division of labor in unicellular organisms, but in multicellular organisms there is a division of function. Lifespan. Unicellular organisms, the cells live short. कहीं ସେମାନେ बहुत गुड़े फंक्शन करती और शीघ्र मरी जाते. But in multicellular organisms, the cell's lifespan is bit larger, is bit more. बुझिला? Okay. एबे आमे टिके आगु को पछु जाय कि पुन थरे देखिबा. Yeah. So under unicellular organism, you may have bacteria. A bacteria is a prokaryote. माने?
It has a non-structured nucleus.
Primitive nucleus. Without a nuclear membrane. Without a nuclear membrane. Pro mane nahi. U mane true. So eukaryote means true nucleus. So it has a nuclear membrane. Bujhela? Second is amoeba. Amoeba is not a prokaryote. Amoeba is unicellular, but it is eukaryote because it has a true nucleus. Paramecium. Paramecium is also eukaryote. Yeast is a microorganisms, but is eukaryote. Jahar nucleus rahila se hochi eukaryote. Jahar nucleus ta primitive without a membrane, nucleoid he kithibo eta hochi prokaryote. क्रोमोसोम टा खोला था तार गोटे कवर किछि न थाए हला. अच्छा, सो एबे हम आगु मूव करबा. ए जो पिक्चर टा तुमे देखुछ, दिस इज बैक्टीरियल कॉलोनीज. दिस मीडियम इज अगर. अगर इज अ टाइप ऑफ प्लांट एक्सट्रैक्ट. आपको बैक्टीरिया ग्रो करिबारे यूज करा जाय लैबोरेटरी रे. लुक्स लाइक अ जेल, येलो जेल. तो ये कौन करागला किछि बैक्टीरिया थिला. एई बैक्टीरिया गुडाक धड़ा धड़ा बॉल भलिया हेकि दिसचंती. एगुडाक गोटे नुह, मिलियंस of bacteria are present at one place. बुझिला? So depending on the nature, unicellular organisms could be free living or colony forming. Free living माने मु एकला बादशाह, मु जोड़े इच्छा जा जीवी जहा करिया कथा करिवि. बुझिला? Colony forming माने गोटे जगह रे ठुलो हेकि रहिबा. बुझिला तो? हां? Why? Because when they are in a colony, they are able to take the nutrients better. They are able to do the work better. For example, bacteria. Bacteria damages your skin. अब केमती करे? Because it releases certain enzymes which damage your cell. अब गोटे bacteria suppose 1 gram करथुओ. Just example दोची 1 gram तो बहुत बड़ा. So 1000 bacteria रही गले 1000 grams बाहरिबो, मतलब 1000 times बाहरिबो. So the destruction would be faster. So this technique is used by pathologists to diagnose diseases. तो सेमाने कौन करंती जते बेले कहार किछि इन्फेक्शन को डाउट करंती. They take a cotton swab. तने तमर थंटीटा जोरे पड़ी गयो अछि. nowadays there is a disease going on, diphtheria. Diphtheria गोटे disease जोटा बहुत दिन धरिकी हो नथिला एबे पुणी थरे चलीची, and it is spreading because people are eating outside, because the spoon, the plate that they use, that transfers the bacteria. So diphtheria, तमर सपोज तार सिम्पटम्स कौन, तंटी पूरा बसी जीवो, ज्वर जोरदार हबो, किछि दिन परे तंटी भितरे मेम्ब्रेन्स तयारी हिजिवो, देयर विल बी थिक म्यूकस, गोटे लेयर है कि तमे ना निश्वास नहीं परिबो ना गिली परिबो, पेन सीवियर हबो. Yeah, around tonsils you have membranes. So this is the symptom of diphtheria. अब suppose you are not sure you have diphtheria, नहीं से धीरे धीरे मेम्ब्रेन बड़ हुए, तमरो ब्रीदिंग प्रॉब्लम कराए, खाइबा पास कराए नहीं, और लोक माने मर जाते, किंतु ता पूर्वरु आजिर समय एंटीबायोटिक नहीं कि लोक भलो होचंती, बुझेला. So suppose the laboratory technicians wants to know कि कौन है, से कौन करिवो, गोटे कॉटन नबो, से कॉटन टाको एमती तमरो पैलेट रे टच कर दबो, जेमती तमे कोविड दोथलो, कोविड सैंपल टच कर दबो कॉटन स्वाब टाको, से स्वाब टाको नहीं कि अगर मीडियम रे रखि दबो. सो अगर होची तार फूड, सो धीरे धीरे बैक्टीरिया ग्रो करिवे. सो जेतेबेले ताकर नंबर टा बढ़ी जीवो, then you can know which bacteria is present, how many, बुझेला. Because in our mouth you don't have only one type of bacteria, you have many types of bacteria at point of time. So if there is a disease causing bacteria, you can correlate कि एई कारण जगो एटा होची, एई बैक्टीरिया जगो एटा होची. Yeah, they test nose samples, but no, the colonies will grow. From the colonies you know what type of bacteria are present, बुझेला का. So एबे पाई एतिकी. I will discuss further in the next class.